and the function of myoglobin as well as collagen. So before going to this class, let me ask someone whether you people are following the lecture or I think some students they are joining, giving attendance and leaving. I'm just downloading the attendance. So let me ask someone. Roll number 282.
Yeah, sir.
So can you just briefly summarize what we had discussed in the previous class?
Sorry, sir. I don't know, sir.
You don't know? You are there in the last class?
Yeah, sure.
So if you are there, you don't know anything?
Yeah, sir. I'm not interested, sir.
Interested?
Yeah.
Interested of what? If you are not interested, you are attending classes, right? You are supposed to go through the lectures. Next, roll number. Roll number 292.
Yes, sir.
So could you please just summarize what we have discussed in the last class?
Sir, I see only the first video. Shall I say that?
Yes?
I read the first video, the first lecture about globular protein.
Yeah.
Sir, from that we study about globular protein structure, function.
Yeah.
When start from globular protein compared to fibrous protein, globular protein is round and sphere structure. It has round and sphere structure. It is functional roles like catalyst and transport, mostly soluble in water, more sensitive to change in pH and heat. That means it changes its 3D structure when affected by pH and heat. Among these, hemoglobin, insulin, and catalase is an example of globular protein. Specifically, we study about globular protein. This is hemoglobin. This protein is found in red blood cell. It is intracellular structure. Human has 270 million hemoglobin molecule in red blood cell. It gives red color for our bloods. It has three major function and property. The major function of hemoglobin is transport oxygen from lung to tissue, carbon dioxide from tissue to lung, regulate pH of the blood. The property of hemoglobin is oxygen binding, oxygen releasing, and cooperativity. This cooperativity occurs when one molecule of hemoglobin subunit bind to oxygen that initiate the next or subcade of oxygen binding of hemoglobin. When structure of hemoglobin comes, it consists of hemi part, that means non-protein part, and globin, its protein part. Hemi part has organic end atoms. Organic consists of protopyrophene system and atom that is iron plus 2 status. This consists of protoporphyrin structure consists of 2 vinyl rings, 2 propionic acid chain, iron molecule bind to 6 binding sites, 4 hydrogen atom. This prevent the conversion of iron to 2 iron state, because when iron 2 convert to iron 3, it is losing the binding of oxygen site. The fifth sites bind to histidine. The sixth sites bind to oxygen. That is important to our blood transport. When we come to globulin parts, which is protein part, consists of 2 alpha and 2 beta chain. 1 alpha chain consists of 141 amino group, and beta chain 146 beta chain amino acid. And also it consists of hemifunctional group, consists of 2 states, R state and T state. R state means redoxed or oxy state. It has high affinity to oxygen, and T states are deoxy state, low affinity to oxygen.
Yeah, good. Yeah. Thank you. I think we'll ask someone else. Harjit.
Yes, sir.
257. Can you continue from there?
So we have seen about the cooperative influences of oxygen delivery in hemoglobin.
Yeah.
The graph about the myoglobin, hemoglobin, which they don't have the hypothetical curve. Then we have seen about oxygenation of hemoglobin, which is accompanied by the conformational changes.
Yeah.
Which it has a histidine F8, and it is associated with residues which are pulled along with iron atom. It also caused the rupture of salt bridges. Then we have seen about the hemoglobin that oxygen and carbon dioxide transports.
Yeah. Okay. Thank you. Next, Ashwati.
Yes, sir.
Yeah, can you continue?
Yes, sir. There's the Bohr effect. That is about the phenomenon that the O2 binding affinity of hemoglobin is inversely related to the concentration of carbon dioxide and H plus concentration. And then there's the CO2 transport. It's transported in the form of bicarbonate, and bicarbonate moves out of red blood cell to maintain the charge balance of the red blood cell membrane, and chloride ion enters the cell from the outside. This is known as the chloride shift, and it reaches the lungs, and CO2 is exhaled from the lungs. Then another molecule that there's a 3 BPG pocket that also.
Okay. Thank you, Ashwati. Thank you. Next, roll number 264. If you are not responding, whether you are in class or should I mark press absent?
Yes, sir. Yes, sir.
Yeah, can you tell me what is the effect of 2,3-ppg on hemoglobin O2 affinity?
Sorry, sir. I couldn't study that.
Okay. Thank you. Next, roll number 300. Bethlehem.
Yes, sir.
What is the effect of 2,3-bisphosphoglycerate on hemoglobin binding affinity towards oxygen?
I do not exactly know that.
Yeah. Thank you. No problem. Next, Ganesh Kumar.
So if the amount of 2,3 BPG increases, I think it decreases the affinity of hemoglobin for
Why?
oxygen.
Why?
So due to the accumulation of 2,3 BPG.
That's not why the hemoglobin affinity towards oxygen reduces when there is a 2,3 BPG.
Because like the oxygen is an amount, right? So need a increased amount of CO2, right?
No, not correct. Next, Jairam.
Can you repeat the question once more? The internet connectivity was a little bad.
What is the effect of 2,3 BPG on hemoglobin affinity towards oxygen?
Sir, I'm not so sure, sir.
So can you tell me what is sickle cell anemia? What we had discussed in yesterday's class. So that means if people are not listening the lecture, or you are taking this course a second time, right? I think you should go through any standard material, or you should listen to the lecture, and you cannot be like this because you need to do well, right, in FAT exam. Next, Balaji Neela.
Yes, sir.
Yeah,
Yes,
what is the effect of 2,3 BPG on hemoglobin?
So I have not reached that part yet, sir. I just started today only, sir, that to revise.
Okay, what do you understood from the previous class?
So we went through about the Bose effect, sir.
Yeah.
And the difference between the alpha and beta chains of the Sir, can you hear me, sir?
Yeah.
Yes, sir, because I'm having little net connectivity issue. Yes, sir, we saw the difference between the alpha and beta chains of normal adult hemoglobin, sir. And then, sir, we had also discussed about other forms of hemoglobin and the graphs regarding for that, sir. Effects of pH on O2.
Yeah. At least, thank you. At least you have given outline what we have discussed. Okay, thank you.
Thank you, sir.
So coming to the next protein that is given in the syllabus is the myoglobin. So myoglobin is oxygen storage protein that we had already explained at the beginning of the proteins lecture. And this myoglobin is a cytoplasmic heme protein, and it is mainly present in cardiomyocytes and also the oxidative skeletal muscle of the vertebrates. And this myoglobin is responsible for the characteristic red color of the muscle tissue, and it has three important functions you can see here. And though it is known that myoglobin is a O2 storage protein in the muscle, but it also they releases oxygen in the muscle during hypoxia or anoxia. When there is a low levels of oxygen, the stored myoglobin would be released. Stored oxymyoglobin releases the bound oxygen. And the third one, it also maintains the intracellular O2 concentration in the skeletal muscle. That means it buffers the intracellular O2 concentration. So these are that means it has O2 storage function, and it is also releasing function during hypoxia or anoxia, and it also, as a result, it maintains the intracellular O2 concentration in the skeletal muscle. And in recent studies, it has been shown that in addition to the oxygen storage and transport in the muscle, and it also removes the scavenges the nitric oxide and other reactive nitrogen species which are known to damage the biological molecules. So that means the myoglobin also has the ability to scavenge nitric oxide and other reactive oxygen species. And it is the you can see here that the myoglobin that is present in the serum. So usually the myoglobin concentration is 20 to 80 nanograms per ml in serum. But when there is a damage to the cardiac muscle, because that is known as the acute myocardial infarction. So in this acute myocardial infarction, the myoglobin present in the muscle enters into the circulation. As a result, the myoglobin concentration rises, and so hence this blood-borne cardiac myoglobin is used as a marker to diagnose the myocardial infarction, or it is also called as a heart attack. So that means the blood-borne myoglobin has the clinical significance. Of course, recently this myoglobin has been replaced with another marker, that is the cardiac troponin. So then coming to its structure and function, this myoglobin, as I already said, that it is a cytoplasmic protein mainly present in the cardiomyocytes and the skeletal oxidative skeletal muscle fibers, and the myoglobin and the hemoglobin both came from the same ancestral gene many million years ago. You can see here that this is the same gene. During evolution, both hemoglobin and myoglobin evolved from the same ancestral gene. And this is the first protein that has been elucidated its structure by the X-ray crystallography by John Kendrew. And as I said that it has it can bind oxygen reversibly like hemoglobin. So that means it can bind oxygen, it can also release oxygen. So that means it can bind oxygen reversibly at the site of the heme. And you can also see here that the myoglobin is a monomer, whereas the hemoglobin is a tetramer, and the structure of the myoglobin is similar to the beta subunit of the hemoglobin. It has some similarity between the myoglobin and the beta subunit of the hemoglobin. And of course, the myoglobin, for the elucidation of the structure of the myoglobin, John Kendrick got the Nobel Prize. And as I said that myoglobin not only stores the oxygen in the form of oxymyoglobin when there is a anoxia or the hypoxia, when low levels of oxygen in the muscle, and the stored myoglobin releases its oxygen from the sarcolemma. This is the muscle here. So this is sarcolemma to the mitochondria for the aerobic respiration. So, and coming to its structure, the whale's myoglobin, here it is sperm whale myoglobin is given. It has 153 amino acid residues. You can see here all these amino acid residues are arranged as 8 alpha helices, and all these alpha helices are labeled with letters A to H. You can see here this is the A helix, then this is the B, and so on, A to H. And these alpha helices are arranged to form a globular structure, and you can see here this is the globular structure, and in this globular structure, the as usual the polar amino acids are present on the surface, whereas the non-polar amino acids are present on the present inside the structure. And then coming to the like similar to the hemoglobin, myoglobin also has the heme part as well as the globin part, and the globin part has 153 amino acid residues, whereas the heme part it has, since it is a only monomer, it has only single heme group, and its structure is similar to what we had discussed in the in case of hemoglobin. So we no need to repeat this structure. So this heme again same, and it is made up of with so four pyrrole rings, methane bridges, and the iron atom is present in the center, and it has the six coordination sites, and the four sites are valenced satisfied by the four nitrogen atoms of the pyrrole ring, and the fifth valence is satisfied by the proximal histidine, and the sixth valence is meant for the oxygen binding. And and this is similar to this one. And here in myoglobin, the heme pocket is wedged between the two helices, that is E and E7 and F8. You can see here these two histidines are present. One is a proximal histidine, another one distal histidine. The distal histidine is also bonded to the oxygen by hydrogen bonds, and you can see here the two helices, that is two helices keeps the heme pocket in the correct position in the structure of the myoglobin. And now this myoglobin has the myoglobin is rich in alpha helix. You can see here 77% of the myoglobin structure has the alpha helix. And as usual, if you see the myoglobin oxygen dissociation curve, it is the hyperbolic, whereas the hemoglobin is has a sigmoidal curve. And so from this also we can understand that the myoglobin has the higher O2 concentration, higher O2 affinity. You can see here that because it is a hyperbolic, and when the curve is moving towards left, as I already said that it has the O2 affinity increases. So compared to from this diagram, you can see here that in order to achieve the 50% saturation, myoglobin requires a lower partial pressure compared to the fetal hemoglobin as well as the adult hemoglobin. So the myoglobin, the O2 dissociation curve for the myoglobin is the hyperbolic, whereas the O2 dissociation curve for the hemoglobin is the sigmoidal, and the myoglobin has the P50 value 2 to 2.6 star, whereas the hemoglobin has the P50 value 2 to 26 star. This we have already said in the last class also. So from this, it is very clear that the myoglobin has the higher O2 affinity compared to the hemoglobin. And so coming to the in the myoglobin, the sixth coordination site, also in addition to the oxygen, there are other potential ligands also can bind. That is a carbon monoxide as well as the nitric oxide. And as usual, when oxygen binding takes place, as usual, the same process that takes place, as we have already said, that the iron atom goes into the Fe2 plus ion goes into the plane. As a result, there is a confer that in turn pulls the proximal histidine residue as well. But since myoglobin is a monomer, and hence it doesn't show the cooperativity, so that's why. So it doesn't show the cooperativity like hemoglobin, and hemoglobin is a tetramer. It shows the cooperativity. And the oxygen that binds to the Fe2 plus, in case of the heme pocket, you can see here the first and second oxygen atoms lies at an angle of 120 degrees to the average plane of the heme. And but when the isolated heme has a many-fold higher affinity towards the carbon monoxide, but when in the but isolated heme in the sense in the absence of the protein part, but in the when the heme is present in the globin part, then the histidine residues, that is the distal histidine residue, hinders the O2 carbon dioxide and the oxygen binding perpendicularly. As a result, the carbon monoxide has the higher affinity towards the heme here in the absence of the globin part. That means isolated heme has the higher affinity, but when the heme is present in the globin part, it has the lower affinity because of the hindrance by the histidine residues that are present near the site. And then, as we already said, that the main function of the myoglobin is the oxygen storage, and it is very well evident that the many marine animals and many marine animals they live for a longer period under the diving animals under the water, because if you look at the concentration of the myoglobin in their muscle is many folds higher than the other terrestrial animals, which terrestrial animals, in the sense these marine mammals experiences a condition called apnea, that is the temporarily holding the external breath. Whereas so that means when these marine animals hold their breath, the oxygen that is stored in the form of oxymyoglobin is utilized used for the locomotor activities during the diving activities. And so if you can see here that the myoglobin concentration is high in case of marine animals or the diving animals. You can see here the seal and the dolphin, and they have the higher concentration of myoglobin. This is in milligrams per the gram of the wet mass of the muscle, and compared to the terrestrial animals, it indicates that the marine animals have the higher myoglobin concentration, and this myoglobin concentration is also well correlated with the dive duration in a way in some species. So that means that the myoglobin stores the oxygen, and during apnea, these stored oxygen in the form of oxymyoglobin is released, because as I already said that it is not only stores oxygen, it also releases the oxygen when during hypoxia or anoxia, and for the aerobic metabolism for the synthesis of the ATP. And this is the evidence that the myoglobin main function is oxygen storage, because the marine animals have the higher concentration of myoglobin in their muscle compared to the terrestrial animals. And next important function is the oxygen buffering. As I already said that during extensive the diving, or the oxygen that is diffused into the muscle, if it is not sufficient for the aerobic to meet the aerobic respiration, or to meet the basal metabolic rate, the oxygen that is stored in the myoglobin in the muscle in the form of myoglobin is utilized. As a result, the oxygen concentration is maintained in the cell. That means it maintains the intracellular oxygen concentration in the muscle. And third one is the facilitated O2 diffusion. As I already said that myoglobin not only stores oxygen, and when hemoglobin carries oxygen from lungs to tissues, near the tissues, the hemoglobin releases oxygen, and this released oxygen is near the sarcolemma, is taken up by the myoglobin, and this myoglobin stores this oxygen in the form of oxymyoglobin, and this oxymyoglobin again carries the oxygen from this near sarcolemma to the mitochondria for the respiration, and this oxygen is utilized for the aerobic metabolism because oxygen is a terminal electron acceptor in the electron transport chain for the synthesis of the ATP. And then this deoxymyoglobin again moves back to the sarcolemma, and it is again take up the oxygen that is released by the hemoglobin here sarcolemma, and it stores in the in the form of oxymyoglobin. So that means it has the storage function. It maintains the intracellular O2 concentration, and it also transport the oxygen from the sarcolemma to the mitochondria in the muscle. And as you can also see here, this is the blood capillary, and this is the muscle membrane here. And so the oxygen that is released by the hemoglobin is normally it is diffuses in during if the has some limitation, and when the cell needs more oxygen, that time the oxygen that is stored in the form of oxymyoglobin is utilized. And so these are the three important functions. In addition to this, as I already said, that recent studies have also shown that the myoglobin also not only the stores oxygen and transport and maintains intracellular O2 concentration, it also regulates it also scavenges the nitric oxide concentration. And it is in recent studies it is given that the myoglobin has the ability to scavenge the nitric oxide by converting nitric oxide into nitrate. It scavenges the nitric oxide. In addition to this, since the nitric oxide is also a signaling molecule, and when the say nitric oxide is required, again this nitrate would be further converted back to the nitric oxide by the myoglobin. That means myoglobin it scavenges, it is also releases the nitric oxide. So this is the these are the other functions of the myoglobin, and it has. And so this is about the myoglobin, the structure and function. In summary, let me summarize briefly once again the myoglobin, and it is a cytoplasmic heme protein, and it is mainly present in the cardiomyocytes and the skeletal muscle fibers, and it has the three important functions. It has the three important so it has three important functions. One is oxygen, one is O2 storage, second one is O2 release during hypoxia and anoxia, third one is intracellular O2 buffering. Intracellular O2 buffering. These are the three important functions. And then we discussed about the structure, and this myoglobin has 153 amino acid residues, or it has a single polypeptide chain, and it has only one heme pocket, and this heme pocket is present is wedged between the two helices. And so these 153 amino acids are arranged into 8 helices, and these are labeled traditionally with the letters A to H. And then so coming to the heme part, as I already said, that all these 8 alpha helices form all these alpha helices keep the heme pocket in a correct orientation, and then it provides a suitable environment. As I already said, that the heme pocket again it has the all pyrrole rings, and the iron is present in Fe+2 oxidation state, and so on. Then coming to the main functions, as I already said, that it has a main function is oxygen storage. It is clearly evident from the literature that the marine animals have the higher concentration of myoglobin in their muscle compared to the terrestrial animals, because in the marine mammals experiences a condition called apnea, that is nothing but the temporarily holding the external breath, And in the absence of the external breath, the O2 that is stored in the form of oxymyoglobin is utilized for the aerobic respiration, for the synthesis of the ATP. So that's why these diving mammals have the higher concentration of the myoglobin in their muscle. And in addition to this, the myoglobin concentration is also more in living animals at a higher altitude. This is the again adaptation. And in addition to this, as I already said, that the myoglobin also has the the myoglobin, the O2 dissociation curve for the myoglobin is the hyperbolic, whereas the O2 dissociation curve for the hemoglobin is the sigmoidal, and the P50 value for the myoglobin is 2.6 star, whereas the P50 value for the hemoglobin is 26 star. So from this, it is very clear that the myoglobin has the higher O2 affinity compared to the hemoglobin, and since it has a single polypeptide chain, it doesn't show the cooperativity or the allostery, whereas the hemoglobin shows the allostery or the cooperativity. And then, so in addition to this, the myoglobin also maintains the intracellular O2 buffering, in the sense the myoglobin always maintains the O2 concentration in the muscle, and because in the oxygen, when the oxygen that is getting diffused from the blood into the tissues, that is carried by the hemoglobin, if it is not sufficient for the aerobic respiration or to meet the energy demand of the cell, in such case the oxygen that is stored in the form of myoglobin is utilized for the aerobic respiration. Then third is it is not only maintains the intracellular buffering, it also transports the oxygen from the sarcolemma to the mitochondria in the muscle for the aerobic respiration. And in addition to this, the myoglobin is also known to scavenge the nitric oxide, and this nitric oxide is a gaseous molecule, and it is a signaling molecule, but the excess of concentration of nitric oxide also can damage the biological molecules. So the myoglobin has the ability to scavenge nitric oxide, and it converts into the nitrate. And again, when the cell requires nitric oxide, and this myoglobin also again converts this one to back to the nitric oxide, and that is used for the as a signaling molecule. So that means this myoglobin not only O2 storage and O2 transport protein, it also regulates the nitric oxide concentration in the cell. So this is about the myoglobin. And coming to the collagen. So collagen, we have already you have already studied about the structure of the collagen, and it is the protein that is fibrous protein that is mainly present in many tissues, and it is abundant protein in the vertebrates, and it is mainly present in the teeth, cartilage, and tendon, skin, and blood vessels. And the collagen, and if you see the structure of the collagen, and it has three helices, and that means three chains, and it is a triple helix. That's why collagen has a triple helical structure. And these so this since it is abundant protein in the body, and it is a fibrous protein, and it is responsible for stability of the tissues and organs. It also maintains the structural integrity of the tissues and the organs, and it also provides the greater tensile strength. And it plays an important role. It also plays an important role in the cell differentiation and polarity and the movement, and it also plays an important role in the tissue and the organ development. And coming to its structure, as I already said, that it has three polypeptide chains. It is a triple helix, and these each polypeptide this polypeptide, each polypeptide chain is in the in the left-handed helix, and 3 left-handed helix are coiled in a right-handed fashion and forms a super helix. And if you see this one, each polypeptide chain has roughly 1000 amino acid residues, and that means if the 3 polypeptide chain has almost 3000 amino acid residues, and coming to these amino acid residues, if each polypeptide chain has several several tripeptide repeats, and these tripeptide repeats are glycine, X, Y, again glycine, and so on. Here X is the proline and Y is the often hydroxyproline or the hydroxylysine. So that means what you see in the in a single polypeptide chain, glycine XY, glycine XY. So this tripeptide repeats many times in a single polypeptide chain. And so you can also notice that, so when you take the weight of, when you take the percentage of amino acids present in the composition of amino acid present in the collagen, 30% of the residues, 30% of amino acids are glycine, and the remaining 30% of the residues are proline or the hydroxyproline. In addition to this, 5-hydroxylysine also occurs, and that is the site for the glycosylation. As I already said that the proline, the hydroxyproline, hydroxylysine cannot be directly incorporated during the protein synthesis, because the hydroxyproline and hydroxylysine doesn't have any codon, but only the proline and proline is connected to the hydroxyproline. In the same way, the lysine is connected to hydroxylysine during post-translational modification, and for that post-translational modification, the conversion of proline to hydroxyproline or lysine to hydroxylysine requires prolyl hydroxylase, lysyl hydroxylase enzyme, and that enzyme requires vitamin C as a cofactor. So that's why deficiency of the vitamin C leads to the insufficient formation of the collagen, as a result to the loosening of the collagen bundles, and leads to the bleeding of the gums. And you can also see here that every third amino acid residue, 1, 2, 3, 1, 2, 3, every third amino acid residue in a collagen polypeptide is glycine, because the glycine has a small side chain, in the sense it has only the H instead of R group. The R group is H here, and whereas other amino acids have the bulky side chains. So when the 3 polypeptide chains in a collagen, when the three polypeptide chains are coiled, 3 left-handed polypeptide chains are coiled in a right-handed fashion, and the glycine residues are pushed into the center. Since the glycine doesn't have the side chain, it gives the less steric clash. That's why other than glycine, other amino acids cannot fit into the center. This you will see in the next slide. And you can also see here that this is the composition of the various different amino acids in the collagen. And as I said that it has every third amino acid is the glycine, and the fourth amino acid the glycine. So therefore, 32.7% of the amino acid glycine, and in addition to this, it also has the proline, and the other amino acids are the percentage of other amino acids is less compared to the glycine and the proline. So when the, and of course you don't have the collagen biosynthesis here. So during the collagen biosynthesis, initially all the 3 individual polypeptide chains are synthesized first, and then during post-translational modification, the proline is connected to the hydroxyproline, and once the polypeptide chains are formed, these 3 polypeptide chains in turn coil in a right-handed fashion and forms a collagen molecule, and this collagen molecule in turn associate and forms a collagen fibril, and several collagen fibrils, which in turn forms the collagen fiber. So this is the structure of the collagen fibers that are present in the various connective tissues, what I have explained at the beginning. So now coming to this one, when the 3 polypeptide chains are coiled in a right-handed fashion, you can also see here there is a intrachain hydrogen bonds between this carbonyl oxygen group and the proline and the glycine NH groups, and that gives the rigidity to the triple helix. And as I already said that the lysine is converted into hydroxylysine, or proline is converted into hydroxyproline only during the post-translational modification. And for that, the hydroxylase enzyme is required. This hydroxylase requires vitamin C as a cofactor. You can also see here, this is the overview of the collagen biosynthesis. And initially, all these polypeptide chains are formed first, then and of course, there are certain signal peptides would be removed during the processing. And once the matured collagen formed, and that again these triple helical collagen further in turn forms the collagen fibers and the collagen fibrils and collagen fibers and so on. These collagen fibers are present here, present in the tissue. And then, as I already said, that when the three polypeptide chains are coiled, three left-handed alpha helices are coiled in a right-handed fashion, and as I already said that the glycine is pushed into the center. And you can see here, since it is a sterically crowded environment, and since the glycine doesn't have the side chain, and so the glycine gives the less steric clash. That's why only glycine can fit into the here. So that is therefore, if the glycine is replaced with any other amino acid, the triple helical collagen may break, in the sense other amino acids may not exactly fit into the center when the three polypeptide chains are coiled tightly. And this is about the collagen. And of course, there are different types of collagens present identified. There are more than 20 different collagens are present in the same in the same individual in different tissues. And the type 1 collagen is the major collagen that is found almost many mainly distributed, like in skin, bone, tendon, cornea, and also the blood vessels. And the type 1 collagen is the major collagen, and of course there are, as I already said, that more than 90 to 20 different collagen types are present, and the type 1 is the major collagen. And the type 2 collagen is mainly present in the cartilage and intervertebral disc, and the type 3 collagen is present in the fetal skin and also the blood vessels. And the type 4 collagen is present in the basement membrane and the muscle. And the type 5 collagen present in the placenta and the skin. And so they have the slight difference in their distribution and also the structure. And the type 1 collagen is the major collagen that is found mostly. And so these since I said that the collagen has a 3 polypeptide chains, and in all these 19 collagen types, these 3 polypeptide chains are either they are identical, or the same polypeptide, they are made up of the same type of polypeptide chains, or they are different. In case of type 1 collagen, and the 2 alpha 1 chains and 1 alpha 2 chain is present. That means 2 chains are identical and 1 chain is different. As I already said that it has each polypeptide chain has 1,000 amino acid residues roughly, and it has many the many repeats of the glycine, X, Y glycine repeats, and the every third amino acid residue is the glycine, and that gives the less steric clash such that the 3 polypeptide chain coils tightly. So this is about the structure of the collagen. And